Hi everyone, and welcome to the next webinar in our new Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I am your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you are experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all the associated materials within the next few days. If you have questions during today's webinar, please let us know using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes for questions. Benjamin Swessinger, who is an ARC Future Fellow and Independent Group Leader at the Australian National University, is with us today to moderate your questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar is the fourth in our new virtual research talk series in response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and to sign up if you'd like to be considered as a speaker. You can find us on social media at plantae_org and at ASPB. For those of you listening to this as a recording, feel free to reach out to us via Twitter with follow-up questions or comments using #PlantaePresents. Thanks again for joining us today. I'm now going to turn it over to Benjamin to introduce our speakers and moderate today's session.
Thank you very much, Katie. So I'm Benjamin. I'm at the Australian National University, a group leader, and we mostly work on fungi. But today we will talk about two very exciting talks about plant development. First, it will be Keiko Sugimoto, followed by Hongtao Liu. I'm really grateful that both could make it today. So first, I will introduce Keiko Sugimoto. She will be talking about molecular mechanisms of cellular reprogramming during plant regeneration. Keiko is a team leader at the Riken Center for Sustainable Resource Science in Yokohama, Japan, very lovely place. She received her Ph.D. from the Australian National University and completed a postdoc at the John Innes Center in the UK. Her research focuses on the cellular control of cell and organ size, cell division, wound healing in plants. Thank you, Keiko, for taking the time today and talking to us.
Okay, so thanks very much to Katie and Ben for organizing the seminar today. And also I want to thank Eugen and Mary for making this possible. I think this is a great idea. And not only for the working from home, but also for the global audience. And I hope this seminar series will continue. We can continue this after we go back to the lab at some point. So I'm going to tell you about the cellular reprogramming today, and I'm going to talk about this in the context of the plant regeneration. And in my talk today, I'm going to take you through these things. So why, that this is, I guess many of you are staying at home right now. So I show you how you can make your life more interesting and entertaining by, spotting or even inducing regeneration at home. And I'm going to tell you why, this is so interesting to study in biology. And then I will also talk about how much we understand right now for the molecular mechanisms of the regeneration. And I want to end my talk by giving you, some ideas about where we should be heading out for. So, regeneration at home. So I guess we're out of the lab and spend lots of time at home, and you realize there's actually a lot of plants that can regenerate very well. I'm just showing you a few examples here. And for example, this one, the lots of the ornamental plants, they are very good at regenerating. So if you cut their stems and stick it onto the soil or water, then within a few days they start making these new roots from these cut sites. Some of you might be out in the garden and then try to dig out the dandelion, because they are beautiful flowers, but they can be also notorious, invading into your space. And the harder you try to remove this dandelion, I can tell you, the more you get the flowers, because you leave out these root segments in soil, and in each of these segments, they can produce new shoots. So the harder you try, the more flowers you end up having in your garden. Another maybe a good example of or maybe the familiar example of the regeneration at home is a grafting, and you see this grafting in lots of vegetables and trees. So here we're looking at the tomato plants here, and if you zoom into this stem, you can see this is actually made out of the two different plants, and one is the rootstock, which is often resistant to soil bacteria or other stress conditions. But then it's fused to the other part of the plant, which is Auxin, and that's to of course produce lots of nice fruits and so on. And you can do this also in autopsies. So the Charles Mernick does lots of nice work using this system in autopsy hypochondros. And I took this photograph from his early work, and you can see from these cut sites or grafting sites. And if you zoom into this, you can see this rootstock, and then there's also a sire, and this is the grafting sites. And you can see this is where cells need to divide, so they get back into the cell cycle and then proliferate, but also to reconnect the vasculature. So this is a beautiful example of the tissue regeneration. And you also see grafting in lots of ornamental trees as well. So I guess the rose starting is very soon, and you see in essentially every rose trees you find on the market right now is grafted. And again, using the very strong resistant rootstock, and then having the beautiful this Auxin that produce nice flowers. They're fused together. So if you really look at this stem area, you might also see these grafted sites. And just one last slide on the grafting. This is not something we really force the plants artificially to do. The plants are actually very good at doing the natural graft. So if you go out to the park or nature for jogging or walk, whatever, and if you pay careful attention, you see these graft sites are naturally occurring in lots of them. So you realize the plants are very flexible, very intimate than we think, and maybe than we are, because this is something we never do as a human, or any other animals would never do something like this. Okay, so why this is interesting? So I gave you a couple of examples of the plant regeneration. Oops, and I gave you, there's different kinds of modes. Sometimes shoots makes roots, sometimes roots makes shoots, and so on. But there's one thing in common in this, and that is about the making of a new organ. Making of a new organ from the cut sites. And if you think about what we do as an animal, this is quite extraordinary, because what we do, or planalia or fish or amphibians, what they do, if you experience the injury, you can repair the tissue that you have lost, but we never start making something completely new. And the other thing that makes the plant regeneration very interesting is because we already know the plant hormones can promote the regeneration a lot. And here we have the Arabidopsis hypochondra X plants, and Arabidopsis, I have to say, is not a very super regenerating plant. So if you just cut part of the X, the tissue and put it on a media, they don't do very much. But if you supply auxin in the media, then they start making these roots, regenerating a lot of roots. So you can clearly see auxin is a huge promoter of the root regeneration. And if you also give, in combination of the auxin and cytokinin now, when you have the right concentration, the right ratio of the cytokinin and auxin, you can see sometimes the shoots are being formed. So the auxin cytokinin can convert this kind of plants into either making lots of roots or regenerating lots of shoots. And that's very interesting why this can happen. Now, another very interesting aspect of the regeneration, something maybe perhaps slightly underappreciated or understudied, investigated, is that not only these plant hormones, but we also need to give the wounding stress to promote the regeneration. And so here we have the unwounded, so intact abscess roots, and we grow this on the hormone-containing media, and they produce lots of lateral roots because we have lots of auxin. Whereas if you give this cutting to the roots, and that's the only time when you start making this shoot regeneration. So that clearly shows you need wounding stress to promote the regeneration. And again, this is a very interesting phenomenon, and we want to understand the molecular mechanism behind this. So if I summarize what I already told you, and then look at this on a more the cellular scale, I told you, we need a wounding stress in combination of the auxin and cytokinin, and some cells in the tissue start responding to these cues, and they undergo cellular reprogramming and also re-enter the cell cycle. I think there's a big issue, which comes first between reprogramming cell cycle re-entry, but they do this reprogramming and re-entry one way or other, or one after another, and then only then we can start making the new organs. So the question, I think we're trying to understand very hard right now is what is the wound stress, and how do plants sense this stress and transduce signal to promote the reprogramming, and also how do plants integrate this wound signaling with the auxin cytokinin developmental signalings to be able to promote the reprogramming. And also, although this is fantastic capacity that plants has, but we don't want this happening all the time. We want to make this happen only at the right time. And the recent studies is really starting to show plants do have a mechanism to limit this capacity, and we are making a very good progress on this. So I'm very happy to see that the regeneration community is really expanding right now, and I think the regeneration research is really happening. And we made a huge progress in asking or addressing these questions. But today I'm going to take you through what we have discovered in the recent years, and then show you where we are. So the whole regeneration study that we do right now in the lab really started when Akira joined my lab some time ago now, and after he found this very interesting regulator we named WIND-1. So WIND-1 stands for the wound-induced differentiation one, and it's an AP2ERF type of transcription factor. And what's interesting about WIND-1 is this gene is rapidly induced after wounding. It comes on within the first hour after wounding. And this slide is showing the WIND-1 activity, the promoter activity after the wounding, 2 days after the wounding. And you can see this is abscess leaf, this is the cut size, and you see the WIND-1 promoter activity is really restricted to the wound sites. So you can see it's a gene induced by wounding. And I'm going to show you a few slides to really show you that it's acting as a key regulator of the wound-induced reprogramming. So if you overexpress the WIND-1 without giving any plant hormones on the media, of course the wild type plants grow very happily, but you can see this WIND-1 overexpressing plants, it's really making lots of callus cells, so they are undergoing the cellular reprogramming. We can also do these experiments after giving the hormones. And now in this experiment, we are not cutting, so we're not giving any wound stress, but we just give the hormone supply. And as I already told you, the wild-type plants do not make any shoots, so it just makes lots of lateral roots because of the auxin. But if we combine this with the activation of the WIND-1, that's sufficient to start making the shoots. And that's very interesting because that really tells you all you need, at least what you need by wounding is the strong induction of the WIND-1. And as long as we give the WIND-1 induction without giving any additional cuts, that's sufficient to reprogram the cell phage and to start making these shoot meristems. We can also do the reverse experiments. So now we're cutting the Arabidopsis expans, and we're even giving the Auxin, which calcium-inducing media, and we transfer these expans to cytokinin, which should inducing media. And you can see the wild-type plants very happy. We're making shoots now. But then if you do the same experiments with the using the WIND-1 dominant negative plants, you see there's absolutely no shoot regeneration happening. And that shows WIND-1 is required for reprogramming the cell phage in this in vitro situation. So I'm not going to go into the detail today, but what we have shown is that after plants receive the wounding, that induces the expression of the WINDs, and there's more than the WIND-1, there's WIND-1, WIND-2, WIND-3, WIND-4. Once they are activated, we have shown that they directly bind and upregulate the expression of a gene called ESR1 for enhancer of the shoot regeneration 1. And once ESR1 is activated, that's also sufficient to induce the downstream genes, and they include a key regulator of the shoot meristem development. So visuals, SDM, COOK1, all of these can come up, and that's the way to induce a shoot regeneration. So this is this one mechanism to show how wounding stress can communicate to the developmental program and to induce the new development, new organogenesis at wound sites. And although I'm not going to go into the detail today, we also show that this pathway, the WIND-mediated pathway, that also can integrate the hormonal signaling. And what we have shown was the auxin cytokinin, they don't really induce the expression of the WINDs, but they can strongly induce the expression of the ESR1 and their downstream genes. So that really shows, we have to have the wounding stress, but having, in addition, the auxin cytokinin, that's really helping to promote this particular pathway. And I like this work because I think it gives us an idea of the earlier points that I made, and this is why and how plants can make this de novo organogenesis at their own sites. And I think it really boils down to the fact that the plants do have the meristem in the post-embryonic development. And you see here we have the shoot apical meristem, and we have the wood apical meristem, and therefore we have these regulators that promote this development. And unlike animals, we don't have this kind of a very highly organogenic perforated tissue. This is where the new shoots are formed and new roots are formed. But because plants do have this kind of tissues, the organs in the post-embryonic development, it might not be entirely impossible to recruit this meristematic program to the wound sites. And perhaps the very clever invention that plants has made was to recruit this pathway and rewire this meristematic program to the WIND-1 or the wound-induced pathway, so that we can ectopically activate this meristematic program right at the wound sites after they receive the wound stress. And so far, I've been only talking about how to promote the regeneration, but I want to switch the gear and I want to talk about in the next few minutes that the plants also have a mechanism to fine-tune their reprogramming. And that's presumably because you don't want this reprogramming is happening all over the place. So we want to make this happen only at the right time. So I'm going to take you through a few works we have done on this. And that's when we came into study the epigenetic regulation. And this is not to say, you know, the epigenetic regulation is all about repressing the regeneration, or regeneration is all done by epigenetic mechanisms. But this is, we can show at least one mechanism that fine-tunes, that limits, restricts the reprogramming. Regeneration is done by epigenetics. And in particular, we're talking about histone modification here. And I'm not going to go into the details of this epigenetic regulation today, but I just want to highlight that in the chromatin, there's two distinct phase or state of the chromatin. And one is the relaxed or open chromatin, and that's where we see the active transcription. But also there's also the region in the chromatin that is condensed, closed, and then that's where you see inactive transcription. And in this inactive transcriptional state, what's very characteristic is that this modification of the histone H3, the K27 trimethylation, when you have this mark on the histone H3, the chromatin region tend to be pretty close, and the regulator that's mediating this modification is the polycomb repressive complex, PRC2. And I'm going to show in the next few slides that some of the key regulators of the reprogramming, they need to be kept in this inactive mode by the PRC2. And when you start losing this PRC2 activity, these genes starts getting expressed, and then that's when you start having the very ectopic unwanted regeneration or the reprogramming events. So this is work done by Akira and Momoko. When she was still in my lab, she just moved on to a new post now to get an independent position. But what they were looking at was this root structure. And these plants also are grown in the absence of the plant hormones. And you see this is a root hair in the white type. These are single cells, and because they end up reduplicate, the nuclei goes up to 16C. But unlike this wild type nice root hairs in the PRC2 mutants, you see these kind of bubbles and bubbles of cells. So when Momoko looked into these structures and see what's going on, what she found using this plasma membrane marker and nuclear marker is that in wild type root hairs is nice, long single cell with a huge nucleus, but in the PRC2 mutants, they are starting to divide. And I have to point out they do form a very nice root hairs, nice and long, but because they cannot maintain this differentiation status, they start to divide. And then after a while, they go really wild, and then they divide quite in a big way. And even from these cows, they start forming this embryo-like structure. So what we have learned from this study was that in the roots we have this stem cell here, and we have the meristem where cells are dividing, undergoing a couple of rounds of cell division, and then they eventually differentiate, and some cells do get into the root hairs. But that's not the end of the story. Even after they complete the differentiation, they still have to actively maintain the differentiation status, and that's done by PRC2. And what PRC2 does is to repress the expression of the very, let's say, dangerous reprogramming genes, our favorite WIND3 or embryonic genes. And when this repression is lifted in the PRC2 mutants like WIND3, ALEC2, they get very strongly upregulated, and that's sufficient to induce the reprogramming, dedifferentiation, and calcification and embryogenesis. So at least one mechanism to keep the reprogramming not happening is done by PRC2. But we also studied how unding permits the transcriptional activation, because we know lots of reprogramming genes, they are upregulated within a few hours after the unding. So unding must be able to create some kind of active chromatin state. And we wanted to know how this happens. So but Ayako and Alice took up this project, and what they've done is to carry out the time course tube sequencing experiments. And we looked at a couple of histone modification, the repressive histone modification and active modification status, and we looked at, after 136 hours after wounding, what kind of changes in the chromatin modification status happening, and what correlates with the gene expression. So here we're showing some examples like WIND-1 that gets upregulated within the first hour after wounding. And what we saw with these kind of genes is that they already actually have the acetylation status before wounding. So they are, in a way, already have this active state that they're ready to be expressed, and they actually express in some cells. But then upon wounding, the expression goes, strongly activated. But we also saw other type of genes and the type of states, and that includes some of these reprogramming regulators. And what we saw was that their expression is really not is close to zero before wounding, and they get strongly upregulated after wounding. And these genes gain acetylation after wounding. So either you already have it or you gain after wounding. And we have shown that this acetylation is very important for wound-induced reprogramming, because if you inhibit this acetylation activity by inhibitor or by mutation, we can strongly suppress the wound-induced reprogramming. So this is the cut sites and the wild type making lots of cell division here, but then there's almost no division happening in this acetylation deficient situation. And we have also shown that this acetylation deficiency is coupled with the reduced induction of the key genes after wounding. So in my talk today, earlier on I showed you the very important transcriptional cascade that links the wounding stress to the developmental program. But in the later half of my talk, I showed you examples of how this transcriptional network is embedded in this epigenetic landscape, so that their expression is modulated in a way that there's no sudden upregulation of the reprogramming genes and unnecessary regeneration, so that it can happen at the right time and at the right place. So that's pretty much the end of what I wanted to show you today, and I just want to highlight, although we made lots of progress, I think we have lots of almost completely unanswered questions, and we have lots to discover in the future. And these are some of the questions we really want to pursue. And although today I advertised how wonderful the plants are regenerating, but there's also a big issue of the recalcitrance, and I think it's very important, an exciting time now that to start understanding or addressing the molecular mechanism behind this not being able to regenerate very well. And then only then I think we can also start finding the methods to improve the regeneration efficiencies. I also want to flash through the last two slides to show that, I talked about the reprogram in the context of regeneration today, but plants can also reprogram in the other context and by other stimuli. And I show you two examples. So it's one nodulation during the symbiosis, and the other is the hostorium formation in parasitic plants situation. And in all of these cases, the cells receive the stress, and they start to reprogram and they start to divide, but then they eventually bring out the very different developmental outcome. And I find it fascinating that, to really see what's the trigger the plants are sensing to initiate the reprogramming, and why and how they can bring out these developmental outcomes. And I think there's lots to be discovered in the future. So that brings me to my last slides. I want to thank my lab, wonderful people, really progressing at the frontier of the plant regeneration studies. And most of the work I talked about is done by Akira and Momoko. The work on the histone acetylation is done by Bart and Alice and Ayako. I didn't have time to talk about what other people are doing in the lab, but let's hope we have another opportunity in the future to share our latest news. So with that, I stop, and I thank you very much for your attention.
Thank you, Keiko. very exciting talk. Regeneration, something so specific to plants and really fundamental to them too. So it's great we're making progress in understanding this important process. And I learned a lot. So I had a couple of questions. We have a couple of minutes for a couple of questions. We will also pass on all the questions which are answered to you, and you might be able to answer them like later on. So there were a couple of questions really around like WIND-1 to 4. So and maybe I just pack them two of them together. So is WIND-1 to 4 functional redundant, or like as a epistatic? And is WIND-1 to 4 also involved in other processes like regeneration?
So I didn't get
Should
the
I
last
repeat?
bit of. Yes, the last bit. Yeah. No, I got the beginning of the
All right,
question, and then. Yeah.
Right. Let me try to repeat again. So the first was about the redundancy and epistasis of WIND-1 to 4. And the second part of the question was, does WIND-1 to 4 also have other roles beyond reprogramming? Because you have like a high baseline expression. So are there any other roles beyond regeneration?
Right. Yeah. So the WIND-1 to 4 is very redundant, and single knockouts do not show any phenotype at all. And we always have to work with the WIND-1 SRDX, which to produce dominant negative. And so I think it really shows it's very highly redundant genes. Whether it works in other processes, I presume so. It is expressed in the pericycle cells and also in the root stem cell area. So it's kind of associated with the let's say undifferentiated state. And although again because of the very strong redundancy, we don't really see development phenotypes even in using the WIND-1 SRDX plants. But I do think WIND-1 do act in the normal development, and in a way the stress pathway is hijacking this WIND-1 function again to recruit this function to the insights and to reprogram.
Thank you. Then going along these lines, there were questions around how well are these winds conserved, and can you kind of explain maybe with the conservation, not conservation in any way, the different plants have different capacities to regenerate?
So WIND-1 is very well conserved. We find them in almost all land plants, I suppose, in monocots, dicots, and also there's very nice work done in the Sugimoto layer, and that's a homologue of, or the cross homologue. There's another EP2 type of transcription factor in the mouse, and they also have very similar function. They're induced by wounding, and they promote reprogramming. So I think the function is very conserved.
So maybe a last one before we have to move on, and then we'll forward you the remaining questions. And of course, you can also ask Keiko questions on Twitter as well with the Plantae present hashtag. So the question is around like what actually does induce the function of WIND genes within these pathways in wounding. So, or if you have herbivory, like what is the actual signaling? Is it like oxylipins or jasmonates, or like what's actually inducing this expression on these whole pathways?
Yeah, I mean, that's the biggest question we're really trying to tackle, what's going on upstream of the WIND-1 induction, right? And I mean, we can only go, up one by one, transcriptional cascade, and try to hit the, the early signal. I mean, earlier studies have shown it's, wounding induced, of course, and of course, in the defense response, there's a lot of the, reactive Auxin species and calcium, induction, and then some older studies have shown these are needed for regeneration. So we're trying to investigate, if these kind of early stress signaling molecules might be involved in upregulating the WIND-1 expression. Yeah, I mean that's really interesting. I mean, the Lin Shu's lab have shown the jasmonate pathway is important for inducing the root regeneration, although we don't really see that in the WIND-1 context. So I think there are multiple ways that the plants sense the wound stress and activate the reprogramming pathway.
Cool. Thank you very much, and thank you for your time and a very interesting talk. If I have more questions, we will pass them on. For now, thank you again.
Our next talk will be by Hongtao Liu, who is a principal investigator at the Shanghai Institute of Plant Physiology and Ecology. Hongtao received her Ph.D. at the Hebei Normal University and completed her postdoc at UCLA. Her research interest lies in the molecular mechanism of light-regulated transcription and circadian clock-regulated transcription, and how light perception controls plant growth and development. Thank you for joining us, Hongtao.
Thank you, Kitty. So, good morning. So today I'm going to talk about how could UVB regulate the plant development. And we know that light is very important for the plant. So not only because of the photosynthesis, but also because of the photomorphogenesis. So light is a very important signal which can regulate the development of the plant. And plants have several kinds of photoreceptors, including the red light photoreceptor phytochrome, blue light photoreceptor cryptochrome, DTL, FKF, phototropism, and also the UVB photoreceptor UVR8. So all these photoreceptors, they work together to regulate the movement responses, growth, and also flowering of the plants. And our lab is basically focusing on the short wavelengths, blue light and the UVB light. So we are focusing on how could UVB light and the blue light regulate the plant development, especially headquarter elongation or flowering. So today I'm going to focusing on the UVB part. I'm going to tell you how could UVB light regulate the development of the plant, especially photomorphogenesis. And UVB photoreceptor UVR8 was first identified from a genetic screening. So you can say that UVR8 mutant shows hypersensitive to UVB stress phenotype. That was in 2002. And later in 2009, people also realized that UVR8 is also involved in UVB-controlled headquarter elongation. But for quite a long time, people didn't know UVR8 actually is a UVB photoreceptor until 2011. So Roman's group, they showed that UVR8 actually is a UVB photoreceptor. So UVR8 form homodimers without the UVB treatment. Well, after the UVB treatment, UVR8 become monomer and accumulate in the nucleus. So UVR8 use several tryptophan to sense the UVB light and then be activated. Okay, and after UVR8 is identified as a UVB photoreceptor, so lots of functions have been found out to be mediated by UVR8. For example, the deethylation, the headquarter elongation, and also the UVB tolerance, the circadian entrainment, phototropism, so on and so forth. So basically, UVR8 mediates multiple UVB responses. But how could UVR8 mediate the UVB signaling transduction? What we know is that UVR8 form homodimers without UVB light, and it become monomer after the UVB treatment, and then accumulate in the nucleus, and then mediates multiple genes transcription. But why UVR8 need to be in the nucleus to be functional? And also, how could UVR8 regulate the transcription? So to answer these questions, our lab we did H2 screening to screen for proteins that physically interact with UVR8. And first, my student Tom, he got BIM1 from his screening. So we find that UVR8 interacts with BIM1 and also BES1, which is a BIM1 interacting protein. So BIM1 and BES1, they are both transcription factors. So they are very important in the hormone brexinosteroid signaling pathway. So BR promoted the activation of those two transcription factors and then promote the elongation of the headquarters of the plants. So it's very interesting that, BR can promote the elongation through those two transcription factors. And now we find that UVR8 can interact with those two transcription factors. And we know UVR8 can inhibit the elongation of the headquarters. So we think that maybe UVR8 can interact with those two transcription factors to regulate the elongation. And we know that BR promotes BES1 by regulate the phosphorylation of BES1. And after be activated by brexinosteroid, BES1 become dephosphorylated. And only the dephosphorylated BES1 binds to DNA. And we find that UVR8 prefers to interact with the dephosphorylated BES1. That means UVR8 only interacts with the BR-promoted, activated BES1. That's very interesting. But a puzzle is that UVR8 interacts with BIM1 and BES1 in a UVB-independent manner. If UVB light can regulate transcription through BIM1 and BES1, and then the interaction is supposed to be UVB regulated. But why? Why the interaction actually is, UVB independent in the yeast cell and also in vitro? So we propose that probably the UVB dependency comes from the relocalization of the UVR8 protein. So we know that after the UVB treatment, UVR8 become monomer and accumulate in the nucleus. So maybe only the nucleus localize the UVR8 interact with those two transcription factors. That means why UVR8 need to be in the nucleus? Just because UVR8 needs to interact with transcription factors in the nucleus. So we tested this hypothesis. First, we did the bifactor assay, and we find that after the UVB treatment, we can see more interaction between UVR8 BIM1 or UVR8 BES1 in the nucleus. And next, we did the co-immunoprecipitation by purifying the nucleus. And we find that after the UVB treatment, we can say that UVR8 really accumulate in the nucleus. And furthermore, there are more UVR8 BES1 protein complex in the nucleus after the UVB treatment. So now we know that the UVB dependency comes from the relocalization of UVR8 protein. So after the UVB treatment, UVR8 accumulate in the nucleus and then interact with those two transcription factors. Okay, so whether BIM1 and BES1 are involved in UVB-controlled headquarters elongation. So those two, they are involved in headquarters elongation is well known. And then we check the ratio between UVB light and only white light. And we find that actually those two transcription factors, their mutants, are hypersensitive to UVB light. That means they are negative regulators of UVB-controlled photomorphogenesis. And our genetics data also indicate that those two transcription factors, they act downstream of UVR8 to regulate the headquarters elongation. Okay, so BIM1 and BES1, they are two transcription factors, and they are involved in headquarters elongation. They can mediate the UVB-controlled headquarters elongation. But whether UVB and UVR8 regulate the BR-promoted headquarters elongation and then to regulate the headquarters elongation. So we check the BR responses of UVR8 mutants. And we find that only after UVB treatment, UVR8 mutant is hypersensitive to brexinosteroid. And without UVB, UVR8 mutant behave the same as the wild type. So it seems that UVR8 really regulate the BR-promoted headquarters elongation, then to regulate the headquarters elongation. And UVR8 overexpression lines, they are insensitive to brachynosterolide, so consistent with the mutant phenotype. So now we know that BR mediates the UVR8 regulated hepcidin inhibition, but how? What's the mechanism? And then we did the transcriptional analysis, and we find that UVB and UVR8 inhibits lots of cell elongation genes transcription, while BR and BES1 promote the transcription of those genes. So UVB and BES1, they work antagonistically to regulate the cell elongation genes transcription. And the mechanism is that UVR8 inhibit the DNA binding activity of BES1. So we did the in vivo chromatin IP and also in vitro EMSA. So we showed that UVR8 interacted with transcription factor B1 and the BES1, and then to inhibit the DNA binding of those two transcription factors, and then to inhibit the BR-induced elongation genes transcription. So our model is without UVB, when you have brachynosterolide there, and then BES1 become dephosphorylated, and then they bind to DNA, promote the BR-induced elongation genes transcription, and then you have a high hepcidin. But when you have UVB light there, and then UVR8 become monomer, accumulate in the nucleus, and then UVR8 interact with B1 and BES1, inhibit the DNA binding activity of those two transcription factors, and then inhibit the transcription of BR-induced genes. Okay, from our screening, we got another transcription factor, WOKI36. So WOKI36 is a WOKI transcription factor, and the function is unknown before. Here we find that the same as BIM1 and BES1, WOKI36 also interact with UVR8 in the nucleus. And we find that WOKI36 is involved in UVB-controlled hepcidin elongation. So it seems that WOKI36 mutants, they are insensitive to UVB. And the reason that they are insensitive is just because even under the white light condition, the mutants show shorter hepcidin phenotype compared with the white type. And after the UVB treatment, they will be shorter, but the ratio for sure is not as dramatic as the white type. So it seems that WOKI36 is insensitive to UVB because it shows constitutively active UVB photomorphogenic phenotype. The reason is that WOKI36 actually is a repressor of transcription factor Hi5. We know that Hi5 is a key transcription factor in the photomorphogenesis. So when you have more Hi5 there, you have a shorter hepcidin. And when you have less Hi5 there, you have a longer hepcidin. And actually, WOKI36 is a repressor of Hi5. So when you have no WOKI36 there, and then you will have more Hi5 gene, and then you will have a shorter hepcidin. And the mechanism is similar as BIM1 and BES1. We found that WOKI36 also interact with UVR8, and then UVR8 inhibit the DNA binding activity of WOKI36 to get rid of the inhibition of WOKI36 on Hi5 gene. So from all these two stories, now we can say that UVR8 accumulates in the nucleus because UVR8 wants to interact with transcription factors in the nucleus so as to regulate transcription quickly in response to the changing UVB light. And also, we find that UVB can regulate the transcription of multiple genes via interacting with different transcription factors. And also, UVB inhibits the transcription of positive regulators while promotes the transcription of negative regulators of photomorphogenesis to make sure when there is UVB light, you will have a shorter hepcidin phenotype. So all these data are about the shoot. So how about the root? We know that for the shoot part, it's easy to get lots of light signal, but most of the photoreceptors are expressed in not only the shoot, but also root. What's the function of the photoreceptors in the root? So to answer how could UVB regulate the root, or whether UVB can regulate the root development, we did an experiment. So we grow the plants on the plate under the white light or under white light plus UVB light. And we find that, interestingly, so UVB light actually inhibit the lateral roots growth and also the development of the lateral roots. So we can say that after UVB treatment, we have less lateral roots there and also shorter lateral roots. And this UVB regulated lateral roots development depends on UVR8. So in the UVR8 mutants, the phenotype is not significant. So UVB cannot significantly inhibit the development of the lateral roots. You may think that, okay, normally the plants grow in the soil, so the shoot is easy to get light signal, but for the root part, so they get very light signal, right? Because it's under the soil. You may think, okay, your experiments were did on the plate, so your root gets a lot of light signal. But you may ask me, that's not the natural condition. You grow the plants on the plate, and then your roots get as much as light signal as your shoot. But in the natural condition, your plants grow in the soil, and then the roots are in the soil, so they get much less light signal than the shoot. So to answer this question, we did an experiment. We grow the seedlings in the soil, and then still treat them with or without UVB light. And then later we dig the plants out, and we find that the phenotype is just as similar as on the plate. So as long as you have UVB light there, you have less lateral roots and also shorter lateral roots. So now we know that even in the natural condition, UVB light really regulate the lateral roots development. And how could UVB light regulate the lateral roots development? So we know that actually auxin is very important for the lateral roots development. So low concentration of auxin promote the growth of lateral roots and also promote the development of lateral roots, while high concentration of auxin really inhibit the growth of the lateral roots. And we want to check whether UVR8 or UVB regulate the lateral roots development through regulating auxin responses. So first, we did the experiment using an auxin responses marker, DR5 gas. And we find that after the UVB treatment, you have less DR5 gas there. And the inhibition of DR5 gas is UVR8 dependent. So we know that UVB inhibits auxin responses through UVR8. And next, we did the experiment. We used high concentration of auxin to treat the plants to inhibit the growth of the lateral roots. And then we treat the plants with UVB light. And we find out that when you have UVR8 there, and then UVB can partially rescue the high concentration inhibit lateral roots growth phenotype. That means UVB light and UVR8 really inhibit the auxin responses. And to further show this, we did the experiment because, you know, when you treat with auxin, I cannot grow the plants in the soil anymore. So to mimic the low light natural condition, so we cover the plates with the foil and then put them into a black box. We get a similar result. So after you treat with UVB, and then UVB and UVR8 can partially rescue the high concentration inhibited lateral roots growth phenotype. So both the experiments show that, okay, UVB actually can inhibit the auxin responses through UVR8 and then to regulate the lateral roots growth. And we also know that UVR8 become monomer and functional and then accumulate in the nucleus. So we want to know, okay, whether the dimeric or monomeric UVR8 is functional in regulating the auxin responses. And our results show that only the monomeric but not the dimeric UVR8 inhibits auxin responses. So only the UVB light activated monomeric UVR8 is functional in regulating the auxin responses. And we know that UBR8 is expressed in both shoot and root. But whether the root UBR8 is functional in regulating the root development, or actually is the shoot UBR8 regulate the root development. To figure out whether the root or shoot UBR8 is responsible for regulating the root phenotype, we did a grafting experiment in Arabidopsis. So we grafted the Y-type shoot, Y-type root, or UBR8 shoot, UBR8 root, or UBR8 shoot and the Y-type root, or Y-type shoot and UBR8 root together. And the result is pretty interesting. So we find that as long as you have UBR8 in the root, and then the phenotype is the same as the Y-type. But when you have UBR8 mutant in the root, even you have Y-type in the root, then the phenotype is the same as the UBR8 mutant. That means the root UBR8 is functional in regulating the root development. So now we know, okay, UBR8 is expressed in the root, and also the root UBR8 is functional, and it can regulate the root development. And to figure out the mechanism, we first did a transcriptional analysis, and we find that UAB really represses the expression of multiple auxin signaling genes. To figure out how could UAB and UBR8 regulate the expression of auxin signaling genes, we did an H2 hybrid screening to find the mechanism. And we find that UBR8 physically interact with MIP73 and MIP77 in a UBR8 dependent manner, even in yeast cells and also in plant. So UBR8 and MIP73 and MIP77, they only interact after the UVB treatment. MIP73 and MIP77 are two MIP transcription factors, and they were reported before to be involved in auxin signaling genes transcription. And we show that actually they act downstream of UBR8 in controlling auxin responses. And the mechanism is that UBR8 interact with those two transcription factors to inhibit the DNA binding activity of them. Okay, now we have the model here. So when there's no UVB light, UBR8 form homodimers, and they cannot interact with MIP77 or MIP73, and they cannot regulate the transcription activity of MIP73 or MIP77, and then they can regulate the auxin responses genes transcription. But after the UVB treatment, UBR8 become monomer and then interact with MIP73 and 77 to inhibit the DNA binding activity of them, and then inhibit the transcription of those auxin responses genes transcription. So now we know that UBR8 can interact with multiple transcription factors, and UBR8 can regulate the transcription through interacting with different transcription factors, and also UBR8 is involved in regulating not only the shoot but also the root part. And it seems that UBR8 photoreceptor actually kind of like a transcription regulator. It can interact with multiple transcription factors to regulate the DNA binding activity of them, so as the plant can respond to the light changing quickly. Okay, so I'm going to stop here, and I would like to thank my team, my collaborators, and the fundings. Thank you so much.
Thank you, Hongtao. That was an incredible talk. Really
Thank you.
All right, so we do have some questions. Let's see. The first couple are just kind of to clarify some of the experimental design aspects. First question was, did you do transcriptomic analysis on isolated roots or the whole seedlings?
Oh, for this one, we didn't do the transcriptome by ourselves. We used the published data. So for the published data, we did compare the whole seedling UVB controlled genes with auxin regulated genes, and also we use the transcriptome feed in root and then compare with the root one with the auxin signaling genes. Yeah.
Okay. And then you had talked about how you had grown some plants in soil alongside some that were grown in media. So you mentioned along the way that some of the plants that you treated with Auxin you weren't able to put to soil afterwards. Why was that?
After we treat the auxin, we cannot put them into the soil. So we cover the plate with the foil and then put the plates in the black box. So to mimic that the plants are growing in the soil, so they get less signal there.
Okay, cool. All right, so now there's a couple more technical questions or kind of more scientific ones to clarify about what you've found here. Can the UBR8 bind all or more than one of the transcription factors that it interacts with? And does it bind at the same time? Through the same part of it?
That's a very good question. So to my opinion, I think UBR8 form different protein complex with different transcription factors. The reason is that for MIP73 and MIP77, they interact with UBR8 actually in a UVB dependent manner, depends on monomeric UBR8. So MIP73 and 77 only interact with the monomeric UBR8. Well, WOKIT36 and BIMO and BEST1 interact with both the monomeric or dimeric UBR8, but they interact only in the nucleus. I think they interact. I mean, they interact in different protein complexes, but we are still working on that. Yeah.
Oh, cool. All right. So next question is: if the UBR8 mutant couldn't perceive UVB, how is UVB making the difference in UBR8 mutants in BR-mediated hypocaudal elongation phenomenon?
Yeah. So this is the slide. So the inhibition comes from the Y type. Okay. So that means after the BR treatment, this one become active and then regulate the transcription of those BR related elongation genes. But in the Y type, and then UBR8 is active, and then UBR8 inhibit the best one. So the growth after BR treatment cannot be very high. But in the UBR8 mutant, because there's no UBR8, so no inhibition, that's why, so the growth is taller. Yeah.
So just a final question is kind of what's next with this work? Like what is your lab, working on and building on with this research?
Yeah, that's a very good
coming
question. So from our lab and also from other labs, we think that there are more than one UVB photoreceptors. So we are trying to screen for new UVB photoreceptors. And also, I think transcription regulation is not the only way to regulate the UVB responses. So we are also working on different mechanisms, such as RNA alternative splicing. Yeah.
Awesome. Yeah, well, thank you. Thank you for joining us today. For those who did attend live, you may notice that this is actually a recording that we did later to overcome some of the audio issues. So thank you so much, Hongtao, for not only doing this once, but doing it twice so that we could get a really great recording out to everybody. I think this is really
Thank you, Kitty. Thank you for your time.
Yeah.
Thank you.
For those of you who are watching the recording, be sure to check out our website, plantae.org. You can go under Education and Plantae Presents and see upcoming speakers and more recordings. So thank you all for joining us today, and be sure to join ASPB to support the series.